Hello, this is Gina Piscatelli with another lecture for Anatomy and Physiology 2 on the intrinsic conduction system of the heart. And this picture on the first slide shows you the anatomy or the elements of this intrinsic conduction system. It's actually a rabbit heart instead of a human heart, but they've used this heart to reconstruct three-dimensionally the components or cells inside the heart that rapidly conduct action potentials or a nerve impulse to ensure that the heart contracts in the proper sequence and with the right timing. So we're going to look at this intrinsic conduction system in more detail. And learn how it helps the heartbeat and the flow of blood through the heart. First, recall that the heart has two different types of muscle cells, or cardiocytes. Some of them are contractile. We call them contractile cardiocytes. And truthfully, the heart is made up mostly of these. So they're the most numerous type of cell in the heart. But there's also specialized cells called conductive cardiocytes that are important for conducting an electrical impulse throughout the heart. These cells are collectively called the intrinsic conduction system. So they're non-contractile cells that generate and distribute electrical impulses throughout the heart. Parts of this intrinsic conduction system are able to initiate contraction without any input from the nervous system at all. So let's first look at the components of the intrinsic conduction system. They include the SA node, which is in the superior region of the right atrium. Then there's a node called the atrioventricular node or AV node, and that's in the more inferior part of the right atrium. Then we have the AV bundle, also known as the bundle of His, and then that bundle branches into a left component and a right component. We just call these left and right bundle branches. These bundle branches extend within the interventricular septum of the heart and then reverse direction and start coming kind of superiorly within the external wall of the heart. Eventually, these bundle branches give off extensions that are thinner, smaller, that reach all the muscle cells that contract in the heart. These extensions are called Purkinje fibers. Now, before we talk about the function of the intrinsic conduction system, let's review a little bit about what we know regarding skeletal muscle contraction versus cardiac muscle contraction. If you recall from A&P 1, the skeletal muscle first needs to be excited by a neuron. So here's an axon terminal that has released a neurotransmitter that binds to a receptor on the skeletal muscle cell. Ultimately, that causes ion influx and a wave of action potentials that travel to the sarcoplasmic reticulum. So this excitation and then depolarization of the muscle cell is coupled to muscle contraction because the sarcoplasmic reticulum releases calcium. And this calcium is needed in order for cross-bridge formation to occur between the actin and the myosin. So the take-home message from reviewing skeletal muscle contraction is that this skeletal muscle first had to be depolarized before it could contract. Okay, so that means it was sitting at a resting kind of negative level, and it becomes a little bit more positive on the inside, causing calcium to be released, which enables the actin and myosin to form these cross bridges and contraction of the muscle to occur. It's very similar in heart muscle. We also have the coupling of depolarization and contraction. The difference is we're not going to have a neuron with a neurotransmitter. Okay, so depolarization occurs first in these nodes. They spontaneously depolarize. It's completely spontaneous, and the SA node does so a lot faster than the AV node does. So it's the SA node that really starts everything, the sinoatrial node. They're considered self-excitable. The cells that are in those nodes are self-excitable. They depolarize and produce an action potential spontaneously. Guess I can't say that too much. And then action potentials are propagated along the atrial walls to the AV node and then to the bundle and the bundle branches. Eventually, the depolarization is going to spread to the muscle tissue of the ventricles. And so you'll also get contraction of the ventricles. And we're going to go through this in a little bit more detail, but this is kind of a big picture. First, you have to have depolarization of the muscle, and then you get contraction. The depolarization is initiated by these specialized conductive cells that spread action potentials to the contractile cells. So the sequence of conduction within the intrinsic conductive system is first the SA node depolarizes, and from there a wave of depolarization to actual contractile cells of the atrium occurs. So action potentials will right away spread to contractile muscle cells, but just of the two atria. But in doing so, some of the action potentials are going to trigger this AV node as well. And so the AV node will then initiate or have its first action potential, which will propagate more action potentials down the length of the AV bundle. And this excitation event occurs then in the branches, the bundle branches, simultaneously, both the left and the right, and then to the Purkinje fibers. So the Purkinje fibers in both the right and the left ventricles will carry this excitation, and that will trigger depolarization finally of the ventricular myocardium. Now, the ventricular myocardium we don't really consider part of the intrinsic conduction system, but ultimately that's where we want the depolarization to go. So this typical rate of depolarization, or the formation of action potentials, is known as the sinus rhythm, set by the sinoatrial node, which is often called the pacemaker. It causes a typical contraction rate of the heart, the ventricles, somewhere between 70 and 80 beats per minute. Now, this can be sped up or slowed down based on autonomic neurons that can impact how fast the SA node depolarizes. So although we consider the sinoatrial node to be self-excitable, spontaneously depolarizing, which it is, the autonomic nervous system can actually fine-tune it. And so if, for example, the parasympathetic nervous system influenced the SA node, you would get a slower heart rate. It would slow down how fast the sinoatrial node starts an action potential. And the same is true for the sympathetic nervous system. It can influence the SA node, and then you'd get a faster heart rate. Okay, let's look at how the SA node establishes its normal sinus rhythm of the heart beating between 70 and 80 beats per minute, or 75 and 80 beats per minute. We need to look at the features of that SA node and figure out how it can actually spontaneously depolarize, just without the nervous system telling it to do so. Well, the SA node, by itself, without any input from the autonomic nervous system, can actually produce an intrinsic rate of action potentials of 100 action potentials per minute. Now, I know before I said 70 to 80, but that's because there's always some parasympathetic input to the SA node. So that's normal. So we normally see that the parasympathetic nervous system is influencing the SA node. But left on its own, if you were able to take out the parasympathetic nervous system, we would see that action potentials in an SA node cell would happen quicker, like 100 times per minute. And so this, notice that this larger diagram on the left shows two action potentials occurring in sequence in a cell of the SA node. Notice that these action potentials look a little bit different than the ones that we studied from neurons. Let's look at the details a little bit, comparing the neuron action potential on the right with the SA node action potentials on the left. First, notice that the action potential in the SA node only reaches a peak of about 0 millivolts, whereas in neurons, the depolarization, the maximum depolarization peak, is at like positive 35. That's one difference. Doesn't explain anything yet, but that's one difference. Another difference is that threshold level is different between the two. So we have a threshold level in the SA node of -40, and we had a -55 in a neuron. That's not that big of a deal, but it's a little bit different. What's really telling, and what is the most important difference, is the resting membrane potential. Notice in the neuron, if you remember, it sits at a resting membrane potential of -70 until stimulated. And it stays at -70 for a certain period of time. The SA node cell is never at rest for very long. It does have a resting membrane potential in the sense that the lowest value is at -60 millivolts, but it spends very little time at that resting. And that is due to the fact that there are always sodium channels open. And so sodium continuously is entering an SA node cell. So it'll slowly creep up as that sodium comes in. That's what makes the SA node self-excitable, is that it's always letting sodium in. Now the sodium-potassium pump is always pumping sodium out, but it doesn't matter, it trickles right back in. So what we've learned so far is that the SA node resting membrane potential is not stable. We say it's unstable. It steadily rises, We call this sometimes the funny current. Again, I don't know where that comes from. And it's due to sodium influx. Okay. Then we've learned that the value needed, or the depolarization level needed for threshold or formation of the first action potential is -40. And now here's another difference. Normally in a neuron, we would then see more sodium influx because more sodium channels would open at threshold. And that's true here in an SA node. But another type of channel opens, and that's a calcium channel. And so you also get calcium influx, which causes a rapid depolarization. So this rapid depolarization is due to two different ions flowing into the cell, both sodium and calcium. Then we learned that the peak of the action potential first generated in the SA node reaches a level of 0 millivolts. The rest is a little bit, well, let's finish at 0 millivolts. What happens at 0 millivolts? Well, since we've reached the peak here, the sodium and calcium channels are going to close. But another type of channel opens, and that's potassium channels. So potassium channels open, and that is just like in a neuron. In a neuron, then, the potassium will leave the So as the potassium leaves the cell repolarizes. It becomes more negative on the inside. So in summary, I want to just show you, if I can, sorry, technological difficulties, hold on. Okay, here we go. In summary, I'd like you to know that the SA node has a slow sodium influx all the time. That's why the resting membrane potential is considered unstable, and that the depolarization for the action potential is due to two different ions, both calcium and sodium. But the repolarization event is the same as in neurons, and that's due to potassium leaving the cell. Okay, let's look at the AV node now just a little bit. So that's what the SA node, we were just talking about the SA node. And it made an action potential, and then one action potential stimulates the next action potential in further along the line of the cell, and then another cell is excited, so on and so forth. Let me just mention here that the AV node is also able to spontaneously produce action potentials or depolarization. But it doesn't do it as frequently, most likely due to the fact that it doesn't have as many sodium channels open. So if for some reason the SA node is not functional, the heart can still be excited, but at a much slower rate. It would be about 40 to 50 beats per minute, the contraction would be. But in a normal situation, the SA node is functional, and it initiates depolarization, which then spreads to the contractile cells of the myocardium and to the AV node. Now, once the action potentials or the electrical depolarization reaches the AV node, the multiple cells in the AV node aren't able to conduct excitation as quickly. So there's a slowing of the excitation through the AV node, and it'll pick up again and go faster once the depolarization reaches the bundle and the bundle branches and the Purkinje fibers. So why does it go slower through? Not so much how, chemically does it go slower, but what's the advantage, right? Well, the importance of slowing conduction at the AV node creates this delay so that the propagation to the ventricles will be delayed. Why do we want that? Because we want to make sure that the atria contract completely to fill the ventricles with blood before they contract. So this AV node, even though it seems like a weird bad thing that it conducts electrical impulses slower, is actually a good thing because it gives time for the ventricles to fill completely with blood before they contract. Okay, I've neglected to talk very much about the spread of excitation through the atria. And I'd like to talk about that a little bit because both atria, the right and the left, contract nearly simultaneously. So what that means is the spread of the excitation to the muscle of both atria is very rapid. So the SA node initiates the excitation, and it rapidly spreads through the myocardium of both atria, of course initiating in the right and moving over to the left. There's a couple of reasons for this, but one that's the most important is that the cells of the myocardium have gap junctions between them. And that is going to allow ions to spread from cell to cell very quickly. And the other feature is that the muscle wall between the two atria, the interatrial septum, is quite thin. So depolarization spreads quickly from the right atrium and throughout the right atrium to the left atrium very quickly. And this is important so that both sides of the heart are functioning simultaneously to fill both ventricles. So blood is going to go from the right atrium to the right ventricle and from the left atrium to the left ventricle. I'm going to show you a picture of these gap junctions. I know it's a little bit cartoony here, but the reason that excitation can spread so quickly through the atrium, as I mentioned, is that there are these gap junctions between cells. You can kind of think of them as channels that allow the ions, sodium and calcium, to spread to the neighboring cell. You don't have to wait for a neuron to release a neurotransmitter and then diffuse across the cleft and initiate the next cell contraction, right? So these gap junctions enable the spread of excitation very quickly. Another feature in the heart that is essential for making sure that the sequence of excitation happens appropriately, in the sense that first the atria are excited, and then there's this nice delay at the AV node, and excitation spreads to the ventricles kind of simultaneously. Another feature that helps ensure that is called the cardiac skeleton. Just think about the fact that if there's depolarization up in the SA node and it spreads through the atrium, what's to stop it from spreading directly to the right ventricle? And that would then be quicker than spreading to the left atrium directly to the left ventricle, right? That would be the wrong direction, right? Because you would get the right side of the heart contracting before the left side of the heart. Instead, we really need the excitation to follow the pathway of the intrinsic conduction system. That is SA node to AV node to the bundles and then up to the ventricles so that they are excited at the same time. Well, this feature that prevents the wrong thing from happening, the cardiac skeleton, is connective tissue that's located between the atria and the ventricles. It's just fibrous connective tissue that doesn't conduct excitation, and it helps ensure the sequence of contraction. I think it's important to note that this is really a miraculous system, not just in achieving the timing of excitation and therefore the timing of muscle contraction, but also it's miraculous in the sense that, or awe-inspiring or whatever, in the sense that the direction of excitation complements the direction of blood flow. So we've already looked at the pattern of blood flow through the heart chambers. And we know that blood flows from the right atrium into the right ventricle and from the left atrium to the left ventricle. That is in a superior to inferior direction. Well, that's exactly the direction of excitation. It starts superiorly and moves inferiorly. So that's how the muscle is going to contract, and that will force the blood top to bottom through this AV valve and into the ventricles. Likewise, we know that blood needs to leave the ventricle or ventricles through both the pulmonary artery and the aorta. And so blood needs to move superiorly now. Because of how, or the direction of excitation moving also superiorly, it complements the direction of blood flow so that you'll have muscle cells near the apex of the ventricles contracting before the muscle cells that are more superior. So it'll like squeeze the blood out from bottom to top. And I think it's really cool to see form following function like that. Okay, that's the end of this lecture, and thank you very much for listening.